So, Nikolaos, if you can share your screen. Nikolaos Chrysanthidis, he is at the KTH in Stockholm, and he's going to speak to us about decoding stimuli in visual working memory.
Hello. Give me 1 second to
No
see
worries.
if it works.
I don't see your screen yet, so we'll wait for the screen to be there. I see it now. Yeah, I see your screen and I see your mouse. So off you go.
Okay, great. So yeah, my name is Nikolaos. Greetings from Stockholm, from Sweden, from KTH. So I'm a PhD student in my second year of my PhD, mainly working with in computational neuroscience area. So what we do, we build large-scale computational spiking models in order to investigate working memory and episodic memory mainly. And more specifically, when it comes to synaptic plasticity, so we combine different forms of synaptic plasticity currently. But interestingly, this project was done under the umbrella of Neuromatch Academy Summer School. The co-authors, Hande is a PhD student at the UCL in UK, and Krishnan was our supervisor in these 3 weeks at the summer school, and she works at the University of Tennessee. So I will mainly present you the project, what was about decoding stimuli in a visual working memory task. So our effort was to predict correctly the stimulation that was used during this task. But let's go to the introduction. So yes, just briefly that decoding information or predicting information from fMRI data has been shown to be quite informative in a range of tasks. And also analyses have shown that different stimulation type that were used during the different memory tasks produced had distinct brain signatures. So these brain signatures could be successfully decoded from bold signals from fMRI. So in this study, we analyzed the published HCP dataset that was given to us. And we mainly focus on the working memory and back task, which included data from a 0 back task, which is a low working memory load, and a high working memory load, which was the 2 back task. So the aim was to determine the stimulus, if the stimulus already can be, let's say, have some different brain signatures by looking already at the behavioral data, then be to predict what was the stimulus type that was used during the, by training a nonlinear model like an SVM with bold signals, and then to see what was the role of specific brain areas in order to decode correctly the stimuli that was used. So the 0 back task and the 2 back task. So in 0 back task, it's just a baseline, very easy task, just to, there are multiple items presented on the participants, but then there's only one item that participants should have it as a target, and each time that this picture is shown to them, they should reply positively that this was the target picture. The 2 back task is a bit more complicated, and here you can see some tools that were used as a stimuli type. And then the participants were asked after 2 steps if the tool that is presented now matches the tool that was presented 2 steps back. And this stimuli is a tool, and it was among the others that we used for this project. So the stimuli that were used in this task was bodies, faces, places, and tools. This is just visualizing the behavioral data. So in the upper figure, we can see the accuracy scores, and the y-axis and the x-axis is the participants ID. So in total, there were 339 participants. And we can already see at the behavioral level that some stimuli types that were used didn't produce very high accuracies. For example, you can see that the blue distribution here, which bodies, different body parts were used as the stimulation types. We can see for high accuracy scores like 100, it's not that dense compared to faces, for instance. And it's a bit more sparser, touching also really low values here. So if we can look now the distribution of times, actually this is a swarm plot, but we already see a shift to higher longer times, for instance. We can see that the participants required more time to answer when the stimuli that was used was different parts of bodies. We can visualize better if we look the distribution of accuracy scores and response times now. So as I said previously, this is now the distributions of accuracy scores. And here we can see when the bodies, when different body parts were used, we can see that the distribution is much wider and has lower values. I mean, this is the plot boxes, and the mean is even lower. And we can see now very clearly that participants required more time to answer about bodies, actually, when body parts were used for this task, which suggests, we ended up that this might suggest that, of course, there is a link between the stimulation, the stimuli types that were used during the task. And as human beings, we are quite social, and we might give more importance, let's say, to faces and places compared to tools and body parts. So this is why we achieve higher performances for different faces and places as opposed to tools and places. So we investigated a little bit further. We did some dimensionality reduction here to bring the bold signals from all the brain areas that we were given to a 2D space to visualize if there is any non-linearity that we could leverage. And here we can see using a t-SNE, actually in the 2D space, we were able to visualize the different clusters. And again, the blue cluster, which is the body parts, is a bit more sparse compared to the other, which is more tight. So this is the PCA. The PCA doesn't produce very good results, but by increasing the number of components, we might also achieve similar results with t-SNE. t-SNE here produces better results because it uses a machine learning algorithm which leverages non-linearity in the data. So what we did then was to train, we used PCA. So we feed the PCA with all the bold signals from the whole brain, and then we produce it 3 components, then up to 360, in order to see if an SVM trained with these 3 components or even more, if we can decode successfully the simulation that was used. So we train a support vector machine with a nonlinear kernel, with an RBF kernel. In the left figure, you can see for low numbers of components, let's say around 3, it's 50-50. It wasn't very accurate, but then as long as we increase the number of components, as long as we increase the information that was used in order to train the SVM, we were able to accurately predict the simulation that was used. So even for 20 components, we were able to predict the stimulation type that was used. It was a body, it was a tool, it was a face with higher than 90%. Then we also did something similar, but from another perspective. So instead of summing up all the information to some components from the whole brain, we decided to to train a DLM, a generalized linear model, and an SVM with specific parcels, with specific signals from specific brains, from specific brain areas. And we can, you can see that we were able to achieve high performance by using only information from secondary visual cortex, as you can see here, which was the parcel ID number 54. And we got high accuracy, like 96% with low standard deviation, both with GLM and an SVM.
10 minutes.
Yes, yes. And yeah, so we're able to get higher accuracy for utilizing this information from specific brain areas. And you can see also the bar plot here, visualizing the differences between GLMs and SVMs. Right, so we expected to see this this high accuracy here because it's a secondary visual cortex and it's directly relevant with the working memory task, as opposed to other brain areas like the somatomotor or network, which is not that active. And by utilizing information, by training the models, having parcels only from this brain area, it's like 50-50. We wouldn't get accurate results. So just to sum up, our conclusions was that we were able to show that there is indeed a connection, a link between the stimuli type that was used and the participants' performances, which is also dependent in the response times. I mean, this is the low accuracy is reflected also in the response times. We were also able to train a simple classifier like an SVM in order to decode successfully the stimulation type that was used during the working memory task, in both working memory tasks. And finally, we were able to distinguish the importance of some specific brain areas like the secondary visual cortex in these working memory tasks. I want to thank you all. This is my contact information. And like I said initially, this project was done during the Neuromatch Summer School, so the Neuromatch Academy is gratefully acknowledged. So yeah, that was.
Thank you, Nikolaos. Great talk. We have 2 minutes for questions. I had a technical question. I was wondering how you were choosing your components. You said that you had the t-SNE, but from there you chose your components?
Yes, I mean, the t-SNE was just to visualize the data in the 2D or 3D. With t-SNE, it's not possible to go beyond that. So we had to use PCA for that reason. But we were able to get similar results with t-SNE, but with higher number of components. So instead of
you use PCA for identifying the components.
Exactly. We used PCA actually to go so we took bold signals from 360 brain areas. We squeezed them, let's say, in 3 components or in 10 components, and then we use this data to train the classifiers in order to, and then we were able to predict.
And you chose them according to their variance, right?
Exactly. Yes.
Very nice. All right. Are there any other questions? If not, then we will slowly transition to our next speaker. Ben, if you can start.